the week of lab practicals. I also want to remind you of the logic of doing that. Lecture is 80% of your grade. Lab is 20. Okay, so if I go up against lab, you better make damn sure I win. Okay, we did axon hillock, we did the branch off the side, which would be called an axon collateral, and then the branches that always occur at the end, which are called axon telodendria, right? Okay, now what are the fingertips called at the very end? Synaptic knobs. That's the most common term. There's a bunch of different names for them, but they're generally called synaptic knobs. So before we get into the function of the synaptic knobs, I want to go back and revisit this myelination thing. Okay, so let's talk about the myelin sheath that exists on some axons. And we already visited this last time. We gave the sort of science fair experiment or science fair idea that said if your copper wire is only that long, you don't need to insulate it to get it to light up the light bulb. But if it ends up going all the way through the house and you don't insulate it, A, you're going to burn the house down, and B, it's never going to get there. So the longer the axon, the more the necessity for myelination. Okay, so myelination, what it actually is, and you can kind of see on this figure that what myelin is one cell devoting itself to wrapping around the axon of this other neuron. Okay, so when you have myelin, what it is it's made primarily of the cell membrane of another cell, so it's mostly made of phospholipids because that's what cell membranes are made of. So let me show you how you form myelin in the peripheral nervous system. So look at here, and then this is going to be how it actually forms. So if you want to myelinate an axon, here's an axon. Where's the neuron cell body? I don't know, up there or down there, but not in the middle, right? So what's going to happen is this other cell, this is one of the glial cells, and it's called a neurolemocyte or a Schwann cell. I don't know if anybody's told you, but in anatomy they're trying to move away from using proper names for things. Unfortunately, that means that you guys will generally have to know both names. So Schwann is the dude who found it, but now they're changing the name to a neurolemocyte. So here's what this cell does. It devotes its entire self and its entire cell life to wrapping around and around and around to insulate a portion of the axon. Okay, and what it ends up doing is pushing the nucleus off to the side and sort of squishing out most of the cytoplasm and other organelles. So it ends up just coating it. And isn't that mostly phospholipid bilayer, if it's just layer after layer? So why does it tend to look white and tend to make tissues look white? Well, if I have one layer of saran wrap, of course it can be translucent, right? But what about a whole roll? That's not translucent. You can't see that, right? You can't see through it. So what happens is wherever you have a lot of myelination, it tends to make the tissue look white, like this, right? So nerves in the peripheral nervous system, they tend to be white or yellowish. And then the white matter within the brain and the spinal cord, that's all myelination. That's all myelinated axons. So it's basically like when you look at white matter, you can tell that primarily what you have is wiring, right? That's what's going on. Okay, so that is how you form myelin in the peripheral nervous system, but there's actually another way to do it. So let me show you. Okay, so the way that you're doing it right here will end up looking kind of like sausage links. Don't you think this looks a little bit like, where are you? Lost you, can't find it. Okay, I just lost everything. Where is it? There it is. Okay, all right, so it looks kind of like sausage links, right? Okay, and little areas that aren't myelinated in the middle, we'll talk about those in just a second. But that's only one way that you can myelinate it. There's another way that you do it in the central nervous system. So let's look at that comparison. So this is what we just saw. This is myelinating in the peripheral nervous system. It's devoting an entire cell to wrap around a part of an axon, leaving little spaces between each cell. This is done with neurolemocytes or Schwann cells, but this is how it's done in the central nervous system. Notice it ends up having the same effect, but it's not actually done the same way. This is done by a different kind of cell called an oligodendrocyte. Oligo means many, dendro means fingers, and cyte obviously means cells. So a many-fingered cell. So what it does is it extends little portions of the cell membrane and wraps around multiple axons, right? But it ends up still myelinating. Okay, so the question is, why the hell do you care which kind of cell does the myelination? Well, it actually has clinical importance because if you damage this axon, these cells, these neurolemocytes will try to help you with reinnervation and fixing the axon. If you damage these, the oligodendrocytes go, I'm out, and they don't help at all, which is why if you damage peripheral nervous system, it's going to be white matter, right? You have the possibility of regeneration. If you damage central nervous system, white matter, you're screwed. Okay, so clinically it actually is important which cell is doing the myelination. So with that in mind, look where we're looking here. What kind of cell is responsible for the color of the white matter in this corpus callosum? Yeah, it's an oligodendrocyte, right? That makes sense? So yet another reason of the 2,000 reasons that you don't want to damage your brain tissue, right? But you knew that before. Now you know a bunch of reasons why. Okay, so Schwann cells are oligodendrocytes. So what does the myelin sheath do? Well, I'm going to give you a little bit of a metaphor to describe what the myelin sheath does so that you can understand it. It's way more complex than this, and it's a little bit of a fib, but it's enough to get you going. So here's what happens with the myelin sheath. So it's an electrical stimulus that needs to travel all the way down the axon, regardless of how long the axon is. And it travels super duper fast along some axons, 250 miles an hour down some axons. But here's what happens. In between the locations where you have myelination, you have these little regions where it's not myelinated. Those regions are called nodes. Nobody can agree on what kind of node, but it doesn't matter because there's only one in the nervous system. They're either neurofibril nodes or nodes of Ranvier for the guy who found them. But don't let it confuse you, because if it says node, that's what it's talking about. It's talking about those little unmyelinated places between the myelination. So here's what happens with those. So what happens at the axon hillock is it's able to create a really strong electrical stimulus. And then what happens in the regions that it's myelinated is it holds on to the electrical stimulus so you can't lose the charge. But then what happens in the unmyelinated region next is it re-energizes the stimulus. So it's going zap, hold, zap, hold, zap, hold. And what that ends up doing is speeding up the transmission down the axon. So it ends up going really quickly down myelinated axons. It's generally faster down myelinated axons. And this sounds like a little bit of a fairy tale, but it's not. There's actually a whole bunch of voltage-gated ion channels at these locations. And so really what happens is wherever it's myelinated, it keeps you from losing the charge, and it almost restarts the charge at every node. And so it goes, they call it saltatory conduction, because those of you who speak Spanish, saltar means to jump, right? But it's not actually jumping. I always think of it as diving, and I don't know what's Spanish for dive. What's Spanish for dive? I want to change the name. What is it? Do we know? Spanish for dive? No? Gosh. What good are you guys? Look it up. Okay, so it's actually going from one place to the next and making it a really fast transmission. Okay, so that's myelination. Okay, so those are your nodes. So now I want to look here, and I'm going to tell you that now you have a lot of knowledge, because if I asked you to, on this figure, and you should do it when you get home, not only could you, of course, label the dendrites and the axon and the cell body, right? You can also label the axon hillock, the axon telodendria, the synaptic knobs. You can tell me which direction information is flowing through this neuron, okay? And if you just thought about it for a second, you could tell me whether that axon is traveling through the CNS or the PNS. Yeah, you can tell it's myelinated with a neurolemocyte, so it's traveling through the PNS, okay? So what happens when you get to the end of the axon? When you get to the end of the axon, of course, you are here at these guys, right? The synaptic knobs. And we know basically what the synaptic knobs are going to do. Structurally, they are going to form a synapse. So what is a synapse? A synapse is the site of communication between a neuron and another cell. It could be another neuron. If that's the case, then it's called a true synapse. Or it could be a nerve cell or a muscle cell. Neurons can command nerve cells and muscle cells too. If it's a nerve cell, it's called a true synapse. If it's a muscle cell, it's called a neuromuscular junction. And if it's a glandular cell, or if it's a gland cell, it's called a neuroglandular junction. That's all in your notes. But what I want to look at is this. Okay, so now you have a decent amount of information in your mind, and you might not even know all the things that you know next. Based on your knowledge right now, not only can you look at one cell and tell which direction information is going, you can look at a chain of cells and tell which direction the information is going, right? So the information, even if the arrows weren't there, the information is going which direction? Right. Okay, that makes sense? That means if there's two cells and a synapse, I could label one of them the presynaptic and one of them the postsynaptic cell, and you would know which one, right? Because the information transmission is unidirectional. That makes sense? So you would be able to label the presynaptic and the postsynaptic cell. Okay, so now let's blow up one of those synapses and talk just briefly about what's going on at the synapse. They look like they're attached to each other, but they're actually not. They're actually microscopically separated from one another. There's a little bit of space, enough to release neurotransmitters in. So if you blow up one of those, I don't test you on this next slide. It's more physiology, but I want to make the physiology sort of relevant to you guys. What's going to happen there is here is your axon telodendria, right? Here's your synaptic knob. Which synaptic, pre or post, is this one? Pre, and this one is post, of course. And then this region right here where they're communicating is going to be the synapse. Why do you care? Well, that's actually the most interesting part of teaching physiology to me, because this is the location where you release neurotransmitters. And neurotransmitters are responsible not just, for instance, for controlling how your muscles function, but also for if you're smart or dumb, if you're anxious or depressed, if you're happy, if you're drug addicted, if you are crazy. All of that is related to which neurotransmitters you released in which quantities and which parts of your nervous system. So when I teach phys, this is a huge chunk of the physiology class. It's a fascinating chunk of the physiology class. In addition, obviously there's a whole bunch of drugs that work here, right? Both fun ones and not so fun ones that work there. And we do a lot of talking about drug physiology in my physiology classes just to make you sort of reinforce normal physiology. Because, for instance, if I told you that, like, whether you release adequate levels of serotonin at your serotonin synapses has to do with whether you're anxious or depressed or pretty regulated on your moods. And what if I told you that there are drugs called SSRIs, selective serotonin reuptake inhibitors, that make the serotonin stay in the synapse longer? You're like, oh, okay, now I get how those work. They're pretending, they're mimicking the effect of me actually releasing more serotonin, right? Or MDMA, which is ecstasy, which actually causes you to dump massive amounts of serotonin, and it stays in the synapse all for a really long time. And you're like, so happy for 8 hours. And then 2 days later, I'm like, I have no serotonin left. Why do I feel suicidal? Because you just dumped it all, right? So this part is phys, and I don't test on this in anatomy, but it's the reason you need a good, strong basis in nervous system anatomy in order to understand the physiology. Okay, so when you look at these two, now, of course, you can label a bunch of different stuff. You've got a presynaptic and a postsynaptic cell. You've got synapses. You've got some dendrites. You've got two different axons, right? You've got myelination. You've got an axon hillock. So you should be able to confidently label all that with just a little bit of practice. And you have learned a tremendous amount about nervous system anatomy already. But you just have to reinforce it by doing that kind of labeling. Okay, so we already talked about the types of synapses that exist. And now we are going to move on to glial cells. So we've just been talking about neurons, and we know a decent amount about neuron structures and neuron functions and what they actually do. And now let's talk about the other things that exist in the nervous system. And the other things that exist in the nervous system are, of course, the glial cells, right? So I'm going to remind you of what the nervous tissue that you looked at looks like. So this is nervous tissue, right? And of course, the neuron gets all of the attention. Not only is it bigger, but it has all of this fancy functionality like electrical stimuli and releasing neurotransmitters. But the glial cells are far more numerous in the nervous system. They actually make up about the same volume. Your brain is about 50% neurons and about 50% glial cells, but because they're so much smaller, there's a boatload more of them. And there's also 6 different kinds of glial cells. So we're going to learn the 6 different kinds of glial cells. Before we get into that, I want to remind you of the metaphor that I used to describe what the hell glial cells were doing. And we already talked about this once before, but so when you think about the neurons that get all of the attention The way that I describe these for you guys to think about is that a neuron is a superstar who's basically been a star, right, since they were a child, like the Olsen twins, right? And because they've been a star since they were a child, they don't know how to do most of the things not related to being a star. They don't know how to put gas in their car. They don't know how they like their coffee. They have no idea what their parents' phone numbers are. They like need an entire support staff or an entourage. And that's how you can think of these glial cells. They all have individual functions, but they're all helping with the functionality of the neuron. And of course, that's important because what if they go bad on you, right? If the Olsen twins at this age lost their entire entourage, do you think that they would function as citizens of this country? No, I don't think they can do anything, honestly. And it's just the way they grow up. Same thing with these guys. If you lose your entourage, you cease to be able to function. So basically the deal is that we're going to have 4 glial cells forming the entourage in the central nervous system. That's like when you're in LA, right? But when you go on location, you only get to take 2 of them. Okay, so there's 4 CNS glial cells and there's 2 PNS glial cells. Does that make sense? Okay, so let's look at the CNS glial cells and talk about what those are like. Okay, so I'm going to use this figure. So we've looked at this figure multiple times, right? Yeah? Okay, so as far as the CNS glial cells, these are the Olsen twins, right? And this is their entire entourage that they're using to help. Okay, but we've already met one member of the entourage, right? We met the glial cells that are responsible for myelination. It's like their communications director, right? So those are the oligodendrocytes, so we already met those. So let's meet the other members of the staff. And the other members of the staff are this guy right here. And the astrocyte, why do you think it's called an astrocyte, just by looking at it? Star-shaped, exactly. So the astrocyte, they used to think was just for structural stability because, of course, it holds on to a neuron and it holds on to something else in the cell. But later on, they figured out that's not all it does. It also is controlling what gets out of this capillary into the nervous tissue. So it's controlling what we call the blood-brain barrier. Have you ever heard of the blood-brain barrier before? I'm going to talk about it in detail in the next set of notes. The blood-brain barrier is a really tight control over what's traveling through the bloodstream and gets into the nervous tissue. Because if you got something funky in there, you don't want it in the nervous tissue because it's amitotic, right? The nervous tissue can't repair. So what this does is really controls access to the neurons, okay? That makes sense? Okay, so that's what the astrocyte does. And then this little guy right here is called a microglial cell for obvious reasons. It's the smallest of the glial cells. What does that do? That's your little badass ninja security guard. He may be small, but do not mess with him, right? So here's the deal with the microglial cells. One of the other things that the blood-brain barrier does, in addition to keeping bad stuff from getting into the nervous system, white blood cells can't get into the nervous system either, which means you don't have the cops on your property. You need an internal security staff. And your internal security staff are these little buggers. And they must work really well because how many of you have had a brain infection? Me neither, right? Okay, so these are little badasses right there. So if anything actually does make it out of the bloodstream into the tissue, they generally whoop up on it so quickly that you never even know that it occurred. Okay, so those are microglial cells. That make sense? And then one last type of cell, and that's these guys right here. But let me show you what those are actually trying to communicate to you. So these are called ependymal cells, and let me show you where they are. So if you blow this up for just a second, do you see that that little section actually got a little corner of this hollow in the brain, this hollow space in the brain? It's called a ventricle. So when you look at what ependymal cells do is they actually line the hollows in the brain. I bet you didn't know your brain was hollow, but your brain is hollow, right? And I'm not even dissing you. Everybody's brain's hollow. Einstein's brain was hollow. So ependymal cells are right here, and here's what these guys do. They line the open spaces in the brain. We will learn the open spaces when we do the brain. They're called ventricles. And the ependymal cells, what they are going to do is suck all of the best stuff out of the capillaries, meaning disproportionate amounts of glucose, the ions that the brain likes, oxygen, and then they are going to dump it into the ventricles of the brain as cerebrospinal fluid. So those cells make cerebrospinal fluid. And I know you've heard of cerebrospinal fluid before, right? Okay, that makes sense? So this is the home team, right? Your entourage when you are at home. And then when you're away, neurons don't have as big an entourage. So when neurons are away, of course, remember when you are away, you are either going to be part of a nerve or part of a ganglion, right? Does that make sense? So in the peripheral nervous system, of course, we already met one of the glial cells. It's called a neurolemmocyte, and it's responsible for myelination in the PNS. Good? And then the other thing that is part of the PNS is that every once in a while you do have some neuron cell bodies in the peripheral nervous system. And when they exist, they're in little lumps called what? Ganglia, exactly. So this is a ganglion right there, okay? If you busted open a ganglion, what would it look like? Inside, it would have neuron cell bodies, right? But then it would also have each neuron cell body protected by a collection of satellite cells. Do you see why those might be called satellite cells? Because they're all around the outside. And what do satellites do? They orbit around the outside. So what are these doing? Well, they're actually controlling access to the neuron, and they're making sure it has the proper amounts of nutrients. So it's basically like an all-purpose personal assistant that you've got there. Does that make sense? Okay, so that's a satellite cell, part of the peripheral nervous system. One last question, just to reinforce the stuff that you learned before. This neuron, this funny-looking neuron, is what structural category of neuron? Yeah, that's a unipolar neuron. And in these ganglia right here, they're all unipolar. Not in every ganglia, but ganglion. Okay, so briefly, let's revisit the concept of glial cells just to talk about a couple of clinical applications. MS, multiple sclerosis, is a disease I bet you guys have heard of. So what happens in multiple sclerosis is your immune system makes a mistake and it starts to destroy the myelination. It doesn't kill off the cell and it doesn't damage the axon. But if you have a really long axon that becomes demyelinated, do you think the signal is going to get through? No, the signal doesn't get through at all. So what starts to happen is you start to have loss of sensation and motor activity, especially in regions where your longest axons exist. So of course you're going to have peripheral effects first, and then they can move in. It can go all the way to the point that you lose control of your diaphragm, and it can kill you, right? Does that make sense? So that's multiple sclerosis. It's an autoimmune disease. There is no cure for it. There's no cure for any autoimmune disease as of yet. That's MS, and it has directly to do with this story. And then the only other thing is cancers. When cancers originate in the brain, because lots of cancers metastasize to the brain, but what about the cancers that originate in the brain? The ones that originate in the brain are far more likely to be gliomas than neuromas, because remember, most neurons are amitotic in adulthood, but glial cells are not. Glial cells go through cell division throughout your life cycle, right? So you're more likely to have a glioma originate in the nervous system. And they even have different kinds of gliomas. You can have an astrocytoma, a microglioma, and that just sort of tells you what kind of cell originated the cancer. Good? Okay. All right. So any questions? Okay. So last thing in the set of notes is just to talk about regeneration of nervous tissue. So if you damage nervous tissue, are you always completely screwed? No, not always. It depends on where it was damaged and which parts were damaged. So first off, we do know that if you damage a neuron cell body, your chances are not good that you're ever going to get it back. Very few neurons in the body actually have the capacity to regenerate. We don't even know how great the capacity is of the few that we learned that actually could regenerate. So at this point, you're just better off not planning on ever getting it back. So if you damage a cell body, the chances are not good. But if you damage an axon, and axons are, of course, far more exposed, right, than cell bodies. If you damage an axon, it depends on whether you damaged a CNS axon or a PNS axon. So if you damage a PNS axon, let's look at what can occur. But then I want you to try to put this together to really think about it. Okay, so I am looking at the peripheral nervous system here. And by the way, let's see if we can do a couple of things. This is, of course, a neuron, right? What structural category is it? Multipolar. And if I tell you this is skeletal muscle, can you tell me more about its functionality? It's a somatic motor neuron. I know it doesn't look like skeletal muscle. It looks like smooth, but then if you blast it up, it's actually supposed to be striated. So this is a somatic motor neuron. So remember something about skeletal muscle fibers for a second. Don't I have to have the nervous system talk to every single fiber? Okay, so let's talk about damaging this one. So if we damage a nerve, let's say we damage the radial nerve, which of course is eventually going to innervate the radial side of my arm, right? We'll have to look at it by an individual axon, and then we'll go back and like expand to a whole nerve. So if I damage this axon, what's going to happen? Well, first thing, this distal region of the axon will disintegrate because it's cut off from the cell body. You're just going to lose it, okay? And then what will happen next, if it's in the peripheral nervous system, is two things. First off, these cells, the neurolymnocytes, will get motivated to go through cell division, and they'll start making a tube. And in addition to making the tube, they're basically secreting chemicals which will attract this little swollen end of the axon that starts to grow again, because of course the cell is still alive, right? So you can make a new axon. So they secrete chemicals which actually courts the axon into the tube. It's called chemotaxis, when you actually secrete chemicals which attract something. So what's going to happen is they're going to try to attract it into the tube. Does it hit the tube every time? No, it doesn't. But if it hits the tube and you have some time to wait, what you might end up getting is complete reinnervation back to that region. But that's one axon, right? And now we have to remind ourselves what a nerve is. A nerve is a bundle of axons. So now I damage an entire nerve, and then the surgeon goes in and puts the ends of the nerve back together. Does the surgeon have any capacity to put individual axons back together? No. And if you wanted complete reinnervation, both sensory and motor, every single one would have to hit the tube and hit the right tube. So what you can get, that generally, by the way, the axon regeneration happens about 1 to 5 millimeters per day. So it can take up to 2 years to figure out whether you're going to get partial reinnervation or total reinnervation or somewhere in between of a nerve, right? So if you had a responsible physician and you had nerve damage, she wouldn't tell you in 2 years you're going to have regeneration. She would say, in 2 years we're going to know what you're going to have, right? And you can't tell what you're going to have because there's no way you can put every single one in the tube, right? Every once in a while, by the way, one of them hits the wrong tube. And you end up with like some weird sensory thing where if I do this, I actually end up feeling it over here. It actually hits the wrong tube and ends up communicating to a different part of your brain. So it's pretty cool. I mean, it's usually in the same region, but if you do this, you actually feel it over here, just like something hits the wrong place. Okay, so that is PNS. What about the CNS? So in the CNS, you don't get regeneration pretty much ever, because if you damage the neurons, they're gone. But then if you damage the oligodendrocytes, they just bug out. And then what moves into the area are astrocytes, which for all practical purposes are just like scar tissue. Because if I was trying to get communication to go from point A to point B, right, and I damaged the wire between them, and then I just had some big yahoo stick a car in between, you're never getting it through, right? So you basically have the nervous system equivalent of scar tissue formation. So you're not going to leave a hole in the brain if you lose the white matter, but you'll fill it with astrocytes, and then you'll actually never get the transmission to go through again. So scar tissue forms from astrocytes. Now, what can we do? Is there any new experimental treatment that actually has the capacity for regeneration of nervous tissue? And the answer is yes, with an asterisk. First off, there's in the lab a lot of experimental treatments going on to try to make an artificial tunnel, right, that will guide the regeneration of the axon. And if you can get it in there fast enough, theoretically, you could keep the scar tissue from forming. But then the other one, the experimental treatment of spinal cord injuries, the other one, I don't know if you guys have heard, you probably have, embryonic stem cell research. So embryonic stem cells know how to become any cell in the body. And if you inject embryonic stem cells into a damaged spinal cord region before scar tissue forms, they actually remember how to make the cells that are in the middle. So they can make you new oligodendrocytes. You'll actually get a new tunnel. Just last week, 2 weeks ago, they had the first spinal cord regeneration in which a man completely, well, not completely, almost completely regained the function of his upper limbs after paraplegia. It was a really weird thing that happened in this country. When George Bush II was president, he decided that we shouldn't do any more embryonic stem cell research because he was very confused and thought that embryos were babies. And so he said that that's like abortion and killing babies. But where the embryos come from for embryonic stem cell research is in vitro fertilization. So in vitro fertilization is about $10,000 a pop. So you don't just generally fertilize one egg and one sperm. You do like 20, right? And then even in Utah, ain't nobody intending to have 20 kids, right? So what do you do after you have the 2 or 4 that you wanted? What do you do with the rest of the embryos? Generally, they just sit in the freezer until they are no longer viable for anything, or you can donate them for research. But he removed all federal funding from embryonic stem cell research, and then we lost all of our scientists. They went overseas. So even though now the federal funding was reinstated, we lost all of our research programs. The only reason that matters is because if you had a spinal cord injury here and the research wasn't going on here, can you fly overseas to get this treatment before scar tissue formation sits in? No, you can't. So most of the research is not happening here. Most of the clinical trials are still not happening here because Bush. I'm from Texas. I can talk about him as much as I want, just so you know. I voted against him twice every time I could. And no, my button is not hooked up to anything, I'm pretty sure. I'm like, hello? Okay, we'll stop there for today. On Monday, bring your brain notes.
So on the picture where you said that it was connected to skeletal tissue, you said that it was obviously a neuron. I'm kind of confused about the difference between a
Neuron and a nerve. This is a nerve, right? A nerve is a bundle of axons, okay? And this nerve would probably be mixed in function because some of the axons would be motor and some of them would be sensory. But with this,
there can be mixed sensory or
yes, but a neuron cannot. So if you look, where is this figure? This guy right here, it's obviously a neuron, right, and not a nerve, right?
because it's not.
Because it's only got one axon, one cell body, right? So that's a neuron. Does that make sense? Okay, so that's a neuron. And then as far as what it's doing functionally, well, it's going to muscle, right? So it has to be motor, okay? And then I said if I tell you this is skeletal muscle, then you can tell me it's also somatic motor because it's going to skeletal muscle, right?
So if
But
that
it can't be
axon-
mixed.
If
Huh?
that axon was bundled, then it can be.
Yes, yes. And it probably is in a nerve, but it's hard to look at the story when you look at an entire nerve.
So the neurons are most likely in the nerve.
No, the axons from the neurons are in the nerves. The cell body is not in the nerve. The cell bodies are elsewhere. Okay?
Thank you so much.
Okay, let me save this and then I'll answer your question.
Thank you. I'm so